Good afternoon, everyone, wherever you are joining in from virtually. I'd like to first thank Children's Cancer and Leukemia Group for the very kind invitation to speak at this really exciting conference. Today, I'm going to give you an overview on a project that was recently very generously funded by the Little Princess Trust and Children's Cancer and Leukemia Group. My name is Ruman Rahman. I'm a research scientist at the Children's Brain Tumor Research Center at the University of Nottingham. And the title of my talk today is Delivering Chemotherapy for Malignant Childhood Brain Cancers Using a Biodegradable Paste. The two childhood cancers that this project is focused on is called medulloblastoma and atypical rhabdoid teratoid tumors, which I'm going to refer to as ATRT for the rest of this talk. So although these are two distinct malignant childhood brain cancers, they share one characteristic in that they both arise in a region of the brain, shown here, called the cerebellum. The image shown here is a magnetic resonance imaging scan, or MRI scan, of a medulloblastoma tumor. Again, this arrow denoting where the cerebellum is. So although 70% of children survive 5 years or more that have medulloblastoma, this is at a devastating expense. And this is because surgery in this very fragile area of the brain carries quite a significant risk of a syndrome called cerebellar mutism. And this is a syndrome associated with temporary loss of speech, reduced coordination and balance. So although these children may survive 5 years or more, they have often lifelong changing disabilities. And these include the speed of processing, affecting motor and cognitive functions. Furthermore, radiotherapy, which can be given to children, particularly children over the age of 3 in the UK, some will receive radiotherapy. This further exacerbates some of these short and long-term devastating side effects. So because of these treatment options, surgery and radiotherapy, which carry such a risk to morbidity, there is a really urgent need to develop more safe and effective chemotherapy treatment regimens. So not only do we want to identify better chemotherapy drugs that would lead to longer survival, but it's just as important that we identify drugs which are safe to use, and in an ideal scenario we could avoid giving radiotherapy because the drugs would be so effective. Similarly, this is an MRI scan of an ATRT tumor in the cerebellum. You can see here this large abnormality is the lesion. This is quite a large ATRT tumor. This is the most common malignant tumor in very young children, those that are under 6 months of age, and it accounts for 20% of cases of children under 3 years and younger. It's a really devastating disease for which there is really no treatment option apart from surgery, and so the median survival is approximately 1 year. The high frequency of ATRT in patients that are younger than 3 complicates the use of radiotherapy. So in the UK, it's very seldom that children under the age of 3 will receive radiotherapy. So for most children that have ATRT, surgery is the only option. So again, there's an urgent need to develop more safe and effective chemotherapy drugs that could not only enhance survival, but to do so in a way that doesn't lead to devastating side effects. Chemotherapy is typically delivered via the bloodstream, and this is referred to as systemic delivery. But unfortunately, most of the administered drug in this manner does not reach the site of the brain cancer. It spreads throughout the whole body. And this leads to toxicity to various healthy tissues and organs which are exposed to the drug. And chemotherapy drugs are very toxic chemicals. And there's no easy way to control the dose that's getting into the brain. And furthermore, the human brain has evolved a barrier called the blood-brain barrier. Normally, this is what protects us from infections and pathogens. It stops these getting into the brain, but unfortunately it also stops many chemotherapy drugs getting into the brain, or it doesn't allow high enough doses for a therapeutic effect. Several years ago, we asked the question that since both adult and childhood brain cancer patients receive surgery as the front line of therapy, could we design something that could deliver chemotherapy drugs at that moment? So this would be where the operating neurosurgeon, after completing the surgical procedure, would deliver chemotherapy drugs directly into the brain. And so the idea is that by delivering drugs in this way, they would target the residual cancer cells that are left behind, that is too risky to remove by neurosurgery because they're penetrated into further into the brain. So we turn to microparticles, or the whole world of very small particles, and in particular a special type of microparticle that makes a polymer paste, which I'm going to talk about in the next few slides. But just before I do, I wanted to give you an idea of the scale we're talking about when I use the word microparticle. So if you can imagine a 1 meter ruler stick, a single microparticle is 1 million times smaller than that. So these are too small to see individually by the naked eye, but when we form a structure, in this case a polymer paste, then that formulation can be observed. So I'm just going to So the polymer that we developed, I'm just going to use the acronym, is called PLGA PEG. It's a biodegradable polymer paste, and it's made up of all those microparticles that I referred to. And if I was to show you the polymer now, if I held up a vial, it would look like a white powder that's free flowing. You could shake it, very much like salt. But if I added one or more chemotherapy drugs in water to the polymer powder, it would create a paste, and it's got a consistency very much like toothpaste. So the idea is that the neurosurgeon at the end of the operation would apply this drug-containing polymer paste to the walls of the surgical resection cavity. The material has been fine-tuned to only stick together and adhere at patient's body temperature, that's 37 degrees, and because it's been applied at close proximity to the residual cancer that is left behind and can't be removed by surgery, the hope is that the chemotherapy would then over several weeks be gradually released and at close proximity. So just a disclaimer that in the next slide I'm going to show a video of a sheep brain that has got this polymer paste. So if you're at all queasy, please do skip the next slide. So this is a sheep brain. The neurosurgeon has removed a portion of the brain mimicking what a brain tumor cavity would look like at the end of the operation. The neurosurgeon has then applied this paste containing the chemotherapy, and then within 10-15 minutes at patient's body temperature, this material has stuck together. And this video is just showing, let me just stop the pointer. This video, when I play it, is just showing that this drug-containing paste has conformed to the irregular shape of the cavity. You can see the neurosurgeon is using a micro spatula just to try and knock any bits off, and you can see that it's kept its shape. So our hope then was that if we deliver drugs in this way, the drugs would seep out over several weeks and penetrate into the brain and hopefully target the remaining cancer cells. It was then important for us to show that this polymer paste does not interfere with the normal brain when it comes to imaging, because patients will have a follow-up image to see if the tumor has returned, and if you had a material that would obstruct the brain signal, then it wouldn't have any clinical utility. So we took that same sheep brain that I showed, and you can just get my pointer again, and you can see here this is the PLGA PEG paste. This is an MRI scan. This is a CT scan. Again, you can see quite clearly that we can delineate where the polymer is from the normal brain signal. So this should not interfere with classical imaging that the patients will receive. So then the most important thing to show was, does this work? And for this, we collaborated with colleagues at Johns Hopkins Medical School in Baltimore in the US, and we used an animal model. They had a rat animal model where the rats had a tumor called a glioma. So this is an adult malignant tumor, and then we tested our polymer paste. And this is the conclusion of that experiment, where you can see here, this is the day's survival in the different groups. So if we gave no treatment to animals, you can see very quickly all the animals succumb to disease. And even if we gave surgery, radiotherapy, and a drug called temozolomide that's given orally, and this mimicked the clinical standard of care treatment, and despite all that, these animals still don't even reach day 40. But you can see here that if we conduct surgery and then deliver the temozolomide and a drug called etoposide via our paste, we have a significant enhancement in surviving animals right up to day 120, and this is further increased with radiotherapy. So this was the first demonstration that delivering drugs in this way can be really effective. We then got the brains shipped back to Nottingham so we could do some further analysis. And where you see very dark purple, this is tumor cells. So in the no treatment animal, you can see quite a large tumor burden as expected. But even in with the animals that have been treated with the standard of care, you can see the tumor has quickly returned and is spreading throughout the brain. But in the animals that received our paste containing both chemotherapy drugs and also then with radiotherapy, we could see no evidence of any residual disease, and this is consistent with the long-term survival of these animals. So this leads us to this project, and now we are embarking, and we've been working this for a year, where we've selected particular drugs, and I'm not going to go into drugs in detail. We have selected drugs that are known to be potent against medulloblastoma and ATRT cells that are grown in laboratory conditions, but when those drugs were trialed in animal models, they were found not to get into the brain. They didn't cross the blood-brain barrier, and we thought, this is the perfect drug candidate for us to test. So the final so we also want to identify new drugs to be delivered via our polymer paste for medulloblastoma and ATRT. So in this project, in parallel, we are developing a model that will help us to understand how brain tumors interact with the normal brain. So the normal brain is the field of neuroscience, and recently one of the hottest topics in brain cancer research is our understanding that the normal brain facilitates the survival of brain cancer cells and promotes the regrowth of the tumor after surgery. So in many ways, the healthy brain is also complicit in cancer progression. So I'm just going to digress, because until now I've been blissfully ignorant to an extent of the field of neuroscience. It's awfully complicated, the study of the human brain, but now we cannot ignore it, and in fact we're incorporating this into our model. But I want to show you how complicated the human brain is. So I do digress here. So this usually it's more fun with a live audience, but let's give it a go here. So I'm going to play this video, and I would like you to count. Let me just pause it. There are two basketball teams, one in white, one in black. When the video plays, they will be passing a basketball to each member of their own team, and I would like the audience to please count how many passes the white team makes. Okay, so if you guessed the answer 13, you're correct. Very well done for your perception. But this time I'm going to just play this video again, and this time don't just sit back, enjoy the video, don't count the basketball passes. And the question I normally ask the live audience at this stage is, who saw the gorilla do the Michael Jackson moonwalk? So let me just play this again. It'll just play for now. Okay, so hopefully you've spotted something glaringly obvious that you may have missed the first time round. The human brain is very complicated. So to model the interaction of the human brain with brain tumor, we have taken a gene from a jellyfish. It's a gene that fluoresces yellow. We've stuck that into normal healthy brain cells, and then we grow the healthy cells with tumor cells in these three-dimensional organoids or mini organs. And then we can use an instrument called the fluorescence activated cell sorter to separate out these two cells, the yellow bright cells from the non-fluorescent cells, which are tumor. And then we can sequence the genome and look at all the genes which are switched on upon this interaction. What are the changes? What are the genes that are switched on or off in the tumor cells upon interaction with healthy brain? And this will give us a clue as to what drugs we can target that would block this interaction and perturb this interaction. And so in this project, in the second phase, when we again will deliver drugs via the polymer paste for medulloblastoma and ATRT, we will identify drugs using this system, this model. Okay, and finally, this is a pre-COVID picture, so it's not up to date, but the individuals here in bold are the three people who are funded off this grant. Dr. Phoebe McCrory, Dr. Carmen Maloney, and Harry Porter has just started his PhD with us, and this is Phoebe here. And this is Stuart Smith, who's the academic neurosurgeon in Nottingham, who conducted all the neurosurgery in the animal models, and the work that we did in the animals and will do in this project is in the labs over at Johns Hopkins University Medical School in Baltimore in the US. So it's really grateful to be able to work with them. And so finally, just to say again, thank you to CCLG and Little Princess Trust for generously funding this grant. And I will be tuning in to the live Q&A session, so please do bring your questions along. Thank you.